Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
All right, Roberto just texted me that he will be a few minutes late. Let's see. Hey, Jerome. Good seeing you. Give it another minute and then we can get started.
Glad to see some of my FDA colleagues are here in good numbers.
Yeah, that's very, very important. So maybe I can start with a brief overview. So this is the continuation of a first discussion and project called Can Artificial Intelligence Grant Challenges Inform Regulatory Science in Anatomic Pathology? Now the first session happened on March, I think it was the 28th, and we wanted to explore how the experience and approaches taken using AI grant challenges could potentially provide data and or knowledge, probably some skills as well that may help to inform regulation and some of the regulatory frameworks that we're working with and under. So the first session contained an overview over the concept, and today we really wanted to take some time to have many of the stakeholders comment and really discuss the topic, and I'm very glad to see many of the FDA experts join as well. Of course, all of this is by no means binding or anything, so we have an official disclaimer on the website. I think people from industry shouldn't discuss territories' prices, so the entire monitoring that generally applies to PI-CC applies to this as well. And any statements made here are of course not specifically authorized by the FDA. This is a collaborative regulatory science initiative, so this is basically a free discussion. I just wanted to state that as sort of a beginning, upfront clarifier that this is, a scientific endeavor, not a, regulatory binding, audience or so. So what I thought is, the two speakers I think will come on. Roberto just said that he is a bit late, but to really hear from the group what sort of the thoughts are, because there are many AI challenges going on, and what people thought about this. And I'm very happy to see many of the familiar faces. So I know that there's been some discussions of even creating a grant challenge, possibly collaboratively with the FDA. Some of the emails that we received were like, how would this even work? Is there a need for a regulatory framework for AI grant challenges that are specifically intended to inform regulation? So there's a broad spectrum of topics. So let's just open it up for discussion. And, as a brief sort of like outlook, I think about halfway through, Brandon Gallis from the FDA, who I'm very happy to see, hi Brandon, wanted to share some of his thoughts. But first of all, let's open this up for a larger scale group discussion. So what do people think of this idea?
Maybe they thought they were prepared comments, Joe, and that this is more of a working meeting and an engagement. So you can hide and listen, that's fine. But the key is really to bring issues forward.
I can make a comment, Brandon.
Sure, Evan.
Kevin from NCI. So I noticed this meeting on my calendar and I couldn't find any link to what it was about or anything. So, and I may have missed the first meeting that that he talked about. But I overall, as you know, I'm a proponent of challenges. I think very that they serve a unique value that other activities so far have not really offered. So, and we're setting up the imaging data commons to be able to work with challenges. The details is to be developed, but there is strong interest from NCI side to garnering challenges as in service of AI development and for cancer research. So I'm happy to help in this initiative. And we have worked with your colleagues on the imaging side, the radiologic side. Like I noticed Kenny Cha is on the call, and others like Nick Petrick in challenges in the past, including pathology. So this is great discussion. Thanks.
Thanks, Kema. Do you know whether any of the image data commons data sets have been used actively in or in support of regulatory submissions?
No, they have not. We would like to, or I should maybe make it more personal. I would like to see that happen. But when I float this idea to my colleagues in NCI, they seem to be in agreement. So we just haven't made a strong push for that. I mean, and IDC, the Imaging Data Commons, as you may know, it's fully public right now, but we do want to have functionality to allow for sequestration of data to activate challenges, etc. And also interested in capturing the artifacts of challenges like segmentations and even algorithms or workflows, etc.
Do you have any thoughts of?
Yeah.
I'm sorry, please.
Just to have a more comprehensive platform and environment for AI data science.
Do you have any thoughts as to why it hasn't been used in the past? Is it more about publicizing, or maybe the content? What is the
why it hasn't been used for what specifically?
For these AI grant challenges in the past, or any regulatory
we have done
missions and things of that nature?
Yeah. So we have not, to the best of my knowledge, on the NCI side, we have not been involved in regulatory science affairs.
Understood.
So that's one reason. The other, why imaging data commons or TCI has not been used directly in challenges because that's not the functionality that they've been set up. So in TCIA, the Cancer Image Archive, we can sequester data. In IDC, currently we don't have, well, actually there is possibility right now, but there's no workflow to address that. So we do want to put that in place. And in services challenges also for network, in-network kind of collaboration, et cetera.
Okay, fantastic. Thank you so much. That's helpful.
Let me tell the group here that public databases have been used in regulatory submissions. I mean, it's not barred or forbidden or anything like that. We have seen them, and in fact, I think the Lung Imaging Database Consortium, something started probably 2 decades ago from NCI, has been used in some submissions, and that's just the one example that I can remember. Because typically when we review something and it's public, we don't like go out and say, hey, someone else used this public database. That's not how we operate. But there's nothing forbidding that.
So I mean, one of the things that was conveyed to us by email was whether an AI data set wants a grand challenge with, let's say, the primary focus to compare, among the participants, to compare the individual performance, whether that data set that seems to be accepted by a certain group of people could form a medical device development toolkit, i.e. a ground truth for, let's say, a regulatory submission to confirm that your specific intended use has been validated on such a data set. Now, I don't know whether any of the existing challenges have thought of submitting that data set as an MDDT, but that was one of the points that I would be personally very interested in what people think about it.
Yeah, we have thought about that too as well. That's actually one of my interests in along those lines to have. And we did have a collaboration, or actually we do have now, with FDA colleagues on SBIR side for contract topics of database as a medical device. I think the applications have been reviewed. I don't think anything has been awarded yet, but that whole concept, our intention is to then bring those data sets to NCI repositories if there is interest from the FDA side as well.
And I'll add, I know specifically of a research project going right now, MIDRIC. I couldn't tell you what the letters exactly spell, but I'm sure the first two are medical imaging data.
Resource Center.
Yep. Well, they are creating exactly that. So,
Yes.
Ivan, you missed that one. They are.
No, I was talking from the NCI side. So you're correct. Okay.
Yeah.
MIDRIC is supported by NIBIB, but the point is well taken.
So that's another kind of NIH-funded initiative to create data sets. That's mostly geared towards COVID-19 and radiology, but yeah, that's just another example.
All right, Roberto.
Okay, thanks, Joe. Thanks, everybody. Coming to your question, Joe, and then relating to the comment of Brandon, I tend to think that the added value of grand challenges for regulatory authorities is actually, and please call me naive, during the clinical trial instead of after or before the clinical trial. As a very concrete example, the Destiny Breast 06 study, the deruxtecan trastuzumab agent for HER2 low breast cancer, and there is emerging evidence that the HER2 low, actually nobody knows what the HER2 low actually is. and there will be big issues once these trials get positive, and there will probably be positive because also they tend to respond even if you have HER2 very low protein on the membranes. And what we need to avoid is that we have the same situation as PD-L1, where we can't go back actually, because the trials have been done, the FDA have approved the assays, they are complying that. It's very difficult to turn the clock back. And if you start too early, the trials will not incorporate this type of knowledge. So my thinking is that let's think loudly. If we have this HER2 low trial, which are being tested in the usual way with a CRO, with the central lab, just a few pathology scoring the HER2. And if we could incorporate within and during the trial a grand challenge using digital tools, so that during the trial in a partnership with the industry, we can collect all this data on comparability of these assays on the trial slides, or even not on the trial slides, but that the assays are being used together with the assay of the trial, so that we have an ongoing validation, analytical validity, and clinical validity of the different assays and the assay used during the trial for that particular trial. So that when the trial ends and the company goes to the FDA, the company has all the information on all those different assays, and then the FDA can decide or advise which assay best to be approved, instead of the usual narrative that there is just one comparative diagnostic that will rule them all. There might be several ones. How crazy is this?
It's not crazy, but the one thing that made me want to add a comment to that is, over the years that I've been at the FDA, you're like, why can't you just use the data from this one company to compare another algorithm? And that's the part that's just never going to fly. We're never going to be able to use someone else's data in that way. Maybe behind the scenes if we had it, but never in a way that other companies could use it. So it really has to come from an outside initiative, whether it's the NCI or a disease professional society or patient group. Those are the people that really have the motivation to create some of these, and even some of the funding to create some of these data sets, and the will and the desire to make it a resource, not just for one company, but for multiple companies. So that's what and even consortiums like Geron's Big Picture Consortium, or that's really hopefully the place where these things will be coming from.
Yep. So I mean, two recent examples are the harmonizing projects in the cell-free, the tumor mutational burden, and the homologous recombination deficiency space. I know I can point to Friends of Cancer Research, who are participants of this regulatory science initiative. I think some of them are on the call today, organizing some of these, let's call it external to the FDA initiatives. Those can provide really important pointers into the right direction. So Roberto, to answer the question slightly different, I think the perceived and undefined value of the data sets created during a trial and upfront handing this over into the public domain is probably the hurdle.
Well, the idea is actually, I understand it. But let's think completely out of the box. So if we look at the experience with the PD-L1 assays in the IMPASSIVE-130, so what happened? IMPASSIVE-130 was done. Atezolizumab was approved with PD-L1 SPL42 as a comparative diagnostic, and that settled a kind of wall around this comparative diagnostic. What happened afterwards is that Roche and Genentech, and we have to again congratulate and commend them and applaud them and throw flowers at them, that they did the comparison between all those different assays on the same trial. But the thing is, they did it after the trial, so after the FDA approval. So it was very difficult, and I understand it, that Roche and Genentech, they couldn't say that there was another assay which was as good as their comparative diagnostic, because what could have happened to the drug label in that sense? So the idea is that this concept, instead of doing it after the trial, that this is being done during the trial. And actually, there are two different ways we could create easily between brackets data sets in which we could test the analytical validity of different assays, including digital ones, but including the assay that is used during the trial for that particular trial of the company. And then we might, as has Genentech done for the Impasse 130 study, comparing all those different PD-L1 assays, to see whether all those different assays have impact on the outcome. And that's an analysis that the company can do. We just give them the essays, and they do it, they perform it on the slides, and we give them the data. They give us the slides, and we do it, and we give them the data, and they do the clinical analysis. So they don't have to share the clinical data with us. And I tend to think that's actually the only way in which we might solve this PD-L1, this companion diagnostic narrative, if that we have to do things like this during the trials, and trials fortunately take years to complete, and we all know that academic projects also take years to complete, unfortunately, sometimes. And I do think that we need to do this type of grand challenges during the trial, and I think the HER2 low idea is actually today probably the most relevant one and timely one, because the Destiny Breast 06 trial is still not yet public, is not yet finalized. And to be honest, I fear that once the Destiny Breast 06 tells positive and the company goes to the FDA, the companion diagnostic will be the assay that has been used during the trial, and we will end up with the same issues again as with PD-L1. Then we will have to implement that particular assay that has been used in the trial. Well, we all know, every pathologist know, that's not the correct way to proceed, because we need to define HER2 low before, and we define which is the assay that defines the best, this definition, before the trial ends. Completely naive, and I know, but I do think that we need to think really out of the box if we want to solve this thing. And we are doing, together with Jeroen van der Schaak's group, these grand challenges on TILs, and we will inform this group about the final results and discuss how we can use this for regulatory purposes, and use the TILs as a kind of paradigm, because it's morphology, on how the FDA might regulate companion diagnostics using digital tools in the future. But we have a real use case today, which is a HER2 low, I think.
Yeah, I think it becomes a question of access, right? So I get your point is sort of, in anticipation of a possible approval, how can we foreshorten the time or the limitations of access to the, to a larger patient population?
Yeah, and I think that's a great target to be shooting for, but I think, that we're not going to be able to do that probably for her too. I mean, the real build up and the culture has to change so that happens, because, there's a lot of forces driving clinical trials, and you can't just inject this force into that whole business right now. But if we keep working at it and, doing some of the stuff after the clinical trial has a lot of value and hopefully gets us to where we want to be too. But in terms of maybe implementing it within the clinical trial, that's a great place to be shooting for. But I think we gotta, when people invest all this money to go into some of those things, you try and keep the covariates limited, you try and keep the patient population limited, because all those things impact the cost of the trial, and so there's not a lot of incentives to really create a new aim or objective for those trials. And so maybe in a future time where the funding mechanisms are just not really aligned with that kind of goal, but the more we try and have some of the real stakeholders, the patients and the health systems, the pathologist societies, kind of driving some of this data generation that the drug companies or whoever can then piggyback on for their clinical trials, that whole switch has to happen in order to have those objectives in the clinical trial, in my opinion. And I'm sitting here, there's no way that this is going to be driven by a regulatory change. There's just too many, constraints, and we're all conservative folks here at the FDA to really and dump over the current working model for something like that, because that's just not under our authority. It's really, that would have to come from Congress, and that probably is a long shot.
Roberto?
Brandon, when coming to the reference material development we have this for immunohistochemistry. We have this for NGS, which we know exactly the variance and the coverage and the variant allelic fraction. We don't have reference materials for digital tools. I was wondering, there is emerging evidence, but it's still very incomplete, but it will become public this year, hopefully, that two different AI tools may be not so concordant at all, and not even concordant compared to the pathologist's scores, but they all predict outcome in the same manner. How do you, and so this suggests that the outcome is probably the, I'm going to say the golden standard, because the moment we say golden standard, we filter out a lot of other thoughts. But to what extent is outcome something that would be important for you to develop reference materials for comparing digital tools, knowing that the concordance between morphology, between the pathologist and AI tools, and between different AI tools is not so wonderful.
So my statistician colleagues have warned me a while ago that we don't even use the word gold standard here at the FDA or truth. We use reference standard, and because there's always some uncertainty. Now, death is pretty certain. I haven't yet to, there's certainly some mislabeling or some documents that might go wrong in taking care of that outcome. But that's a hard outcome for sure. And when I say hard, I mean strong, that we would love to see. But most of the world doesn't have time and the follow-up resources and tracking, and it just takes a lot more effort and money to use those kind of hard outcomes. Now, in a lot of spaces, we do use morbidity and mortality as outcomes for devices. Diagnostics, though, is one of the harder ones because all these things happen on a longer time scale. But that would be a great and wonderful reference standard for any diagnosis. But in order to get things moving faster, we try and be flexible to allow for softer reference standards. In radiology, the reference standard is pathology. And for the longest time, we've been pretty happy with that and haven't really challenged the variability of those pathologic and evaluations. And but then, like in breast cancer diagnostics, there's always been a one-year follow-up as well. So there's always that trade-off between how much effort does it take to get the truth that you want, or the gold standard that you want, and what can we all live with to get things through the regulatory process. And, so that kind of data is wonderful when you can get it.
Well, just to continue thinking completely out of the box and looking where can we find opportunities to find a new narrative and a new framework. There is this, one of the good things about FDA approvals is that you have this conditional approval concept. So you approve something conditionally, and in the meantime, you demand a lot of classically clinical parameters like outcome and the complications and things like this before you grant definitive approval. To my knowledge, and I have searched a bit, I couldn't find anywhere, but it's maybe me, any reference to optimization of the assay or comparability of different assays during the period of a conditional approval and a definitive approval submission. Do you think that would be a window if during the trial, the idea which I posted 50 minutes ago might be an opportunity to consider in this window between the conditional and the definitive approval?
Well,
that
I'm
takes
really
years
not
also.
familiar. I'm not familiar with any conditional approvals besides the emergency use authorization business, and that was a pretty big condition. You know, having a pandemic going on. You know, and there's also some conditional ones because of rare diseases and things. But our business is not really conditional. Once we make decisions, we are not really have mechanisms to go back and change things. Because you can imagine how many lawyers would get into that discussion. So and devices are constantly being changed. I mean, I see diagnostic devices that are on their 30th supplement, you know, over a decade or more. I've seen it on AI devices. I have a little case study on one of the very first breast cancer there wasn't called artificial intelligence, but there's software as a medical device. And over the course of a decade and a half, they had 30, 40 supplements generalizing to new scanners, generalizing to digital. It was started out as film. The film was put in a digitizer, and the CAD was put on, the algorithm was done on the digitized film. And then, you know, in the next 3 years, it was then done on digital images, and then it was even done on 3D. So over the course of 15 years, you know, this thing generalized to all those different platforms. And if there was some forces to drive some of the in vitro diagnostics to play better with other measurements, you know, to be honest to some actual reference standard, you know, developed by NIST or some society that was maintaining it. I mean, that's the thing is, this is probably, you know, we don't have enough resources for our day-to-day job here at the FDA. This has to come from these other outsourced organizations to create these reference standards that can grow. And that's where MIDRIC, the NIH-funded project for COVID-19 data for algorithm testing, is a great example of something that started under the NIH and is turning into a service for AI in that space. And of course, NIH funds a lot of things, and there's a lot of other funders out there, especially patient organizations. And but another group that I've seen make great strides is American College of Radiology with their Data Science Institute, where they're actually part of that MIDRIC project. So I think that's where they thought that they could move their energies, because it also had a little support to go with it, and for a very real pressing issue, that being COVID. So
I know one of the big pushes in the MIDRIC study is, of course, the quality control, right? Because, you know, most of the grant challenges are set up by a group of invested individuals, and I mean, you know, Kevin knows probably very specifically what is talked about when saying moving something to an officially release of a data set, right? There's so many quality issues that have to be considered. Inherent biases, you know, skewing of data because of selection of top quality. So a lot of people criticize TCGA for all that it's worth, for having to select, you know, appropriate materials to do these comprehensive studies. So I think that's a huge challenge in itself. But what do people think about sort of setting up one of the challenges with the specific intent for subsequent, you know, use of that data, akin to what Roberto suggested, for a specific use case possibly related to a clinical trial?
Maybe I can chime in. So if you had the data and wanted to bring it to NCI, I'm assuming this is cancer related, we would find a way to get it to the repositories and be happy to work on a challenge. We also have access to challenge platforms. FDA has also a system. So that's all doable. Do we have data sets?
And Roberto, I'd say the Tiger Challenge is pretty uniquely poised for being used in a regulatory submission. All the stuff I've seen on the challenge website is, you know, it was a little thin in terms of the things that we would look for at the FDA, but I bet you have the information to make it thick with the kind of characterization that we would ask for generally in a submission. And I think the more people go into some of these challenges with that in mind, the more it could be prepared properly up front.
I was going to ask a question to Jeroen, but I saw that he raised his hand already.
Yeah, Jeroen.
Okay, go ahead.
Yeah, I would be interested to hear, Brandon, I'm not sure if this is the place to do that. What you consider thin and what would make it thicker? That would be very interesting to understand from your point of view.
Some of the easiest ways to get some of that information is from some CAD guidance documents in radiology. I think they're great examples for any model building and testing. And I'd be happy to share those and make those go around. But I also, you know, I talk to people all the time that are thinking about submitting an algorithm and as part of my job, and I basically point them to guidance documents that we already have. And I have my little personal cheat sheet of guidance documents and resources that I've said at ICC before, but I'll try and get a link to it in the chat here. And that's, I have just a little advice page with pointers to all guidance documents that I've encountered and believe are relevant, as well as presentations by myself and my colleagues in this space, and also pointers to some of the landmark devices that kind of are the predicates for others, you know, the examples for others. And look, I think someone put that link in there for me. Thanks, Joe. You already knew which one I was talking about. And
of course.
So, and then another nice resource was the actual first alliance paper that Joe led. There is a great, I guess, timeline of all the guidance documents in this space that I think is a great reference for anyone who's getting started in this business to really understand the elements. But from my review, because I review these from the evaluation of the algorithm, I also look at the how these algorithms are built and what their architectures are and, you know, their engineering pieces. But when I think about the key elements that I always have questions about and are usually the weakest part that I want more about is the reference standard. Did you just ask one pathologist to mark it up? Or was there an actual document with instructions and training and, you know, multiple people so that we can average over some of that pathologist variability going into establishing the reference standard? So unusually when we're all said and done, there's at least 5 pages and maybe more on that topic of establishing the reference standard. What are their credentials? You know, those things are the things we look for. What imaging systems are these data being collected on? You know, how many from each different manufacturer? How many different sites? All of that stuff, whereas most grand challenges just say there's 300 images here and they were marked up by one person and go. And that's what I mean by thin, is more depth, more, you know, how many pages do you think a statistical analysis plan should be? Most ground challenges is 1 paragraph, right? And and I would think you would have your before study protocol identifies your target size, you know, and rationalizes why that's the target size. And I've got my little cheat sheet on the side here, but this is a presentation I gave. I don't know if Joe is allowing screen sharing.
Yes, you can.
But this will help me remember some of the topics that I pick out. Are you seeing a slide?
Yep, core content of submissions.
Yeah, and so this is something on that device advice page. I gave this at I think it was ACR, American College of Radiology presentation. And one of besides the reference standard, you know, I left this just one bullet, but that's there's so much to go into that, or could go into that. And I mentioned a few of the issues there. Another thing that is always thin is really clarity on the patient population. You know, inclusion, exclusion. How did you know, I'm sure there were some problem cases along the way. You know, that has to be documented. There might be some convenient populations that you drew in. All that just needs to get documented. And those are the key elements that we always look for. And I've got boxed up here the databases. And a lot of what I'm just saying is, you know, good documentation on where that data came from and what's in that data. Another thing that's always pretty slim, because algorithm developers don't really think of this stuff unless they're in a really good team, right? The really clinical implementation of AI and ML needs the clinical side well represented, and the real clinical context and how does it fit into the current patient workflow needs to be real clear, so that we, you know, just to give us an algorithm loves to give us the probability of disease, but is that actually scaled to the prospective clinical population? No, that's just some artifact of the algorithm at the end. It gives you a magic number between 0 and 100. That probability, is that really linked to the population? You would have to do a prospective, maybe not as prospective trial, but at least you would have to understand the prospective population. And before we would believe it's an actual probability. And so I often go into some of these things. All I want you to do is change the language. Don't call it a probability, call it an index, because that's what it is. Until you prove to me that it's a probability for that population. So this is my go-to slide for elements of a submission. And the more that can be created ahead of time for a grant channel, or even after the fact here, I know Roberto we've been talking a lot on this topic, I think, and he has not only the kind of public challenge where the testing set is private still, there's also a private challenge that is on completely different data that is much more geared towards clinical outcomes. And to me, you know, I think that this is a good opportunity to try and follow this idea through to the end, with or without it being an FDA medical device development tool. You know, when I said we've seen public data sets and submissions, that's true. So what would be nice if it was a tool, then the manufacturers or developers could come in and use that data, and all the regulatory review of that data would happen up front before. And then the idea is that data is of regulatory grade, regulatory quality, the characterization, all this information. And from a manufacturer standpoint, they just need to run their algorithm, produce the performance results, and quote the tool. I mean, so that to me has a lot of value from the FDA side too, because if we review this data once, it could support multiple devices without having to look at it again. The review part takes a while. So the tool, establishing, qualifying it as a tool takes a while. It's a heavy lift.
I think it's important, Brandon, to realize where we are coming from with these challenges. Of course, the goal has never been to go past FDA or to come to you. Of course, we started doing this for comparing different machine learning approaches on the same set of data. And I think over the years, we have improved quite considerably here. And we have challenges like our Panda challenge, which was published last year, in which I think quite a number of these can be ticked, but still it's really great to hear this and to see where we can improve, because sure, it's always, you're always under budgeted, and it's also not always clear which direction to take. So I think it's really helpful to think along these lines as well.
The Panda Challenge was a solid research study. Not, definitely the grand challenges have evolved, and I think now we're at the point like, wait, why are we just using this to play around with algorithms? Let's have some clinical impact. Let's get this stuff on the market. And I think that, I mean, that's what my project is all about. It's not so much embedding it in a grand challenge, although my assistant, Victor Garcia, is ready to offer some of our pilot study data into the challenge, and that would speak to some of the clinical efforts or the private challenge that Roberto has in the Tiger Challenge. So, Victor, do you want to make that offer yourself?
Sure. Yeah, we were looking at the Tiger Challenge, and one of the components is calculating a TIL score as part of, I think it's leaderboard 2. And we have our annotations from our expert panel on our new training material. And I was even thinking if those could be samples to help algorithms assess if they're able to calculate a proper TIL score for their training prior to their submission on the leaderboard.
So we could figure out how to integrate that in. It's not yet to the testing piece. I think our pilot study is lacking on some of the things on the screen right now, because it was a little bit of a convenient set of slides and ROIs. Yeah, I think we're ready to turn this corner, personally.
Joe, can I come?
Yeah, absolutely, Roberto.
I have a few things to add, to complement. So we have indeed these two challenges that we have developed with Jeroen's group and Francesco. In the private challenge, what we're going to use is as training, I'll call it reference materials or training materials, 160 slides that have been scored by 60 pathologists in total, 60 in total, in which we have published already the concordances and the pitfalls on those slides. So that's one set. You have the other one, which is going to be used for the public challenge, the Tiger Challenge, which is a set of 94 breast cancer cases, which I have scored the TILs as such. And other people have annotated the slides, and that's the training material which is publicly available for the groups. And then you have the efforts that you, Brandon, and Kate and Victor have been leading over the past 2 years, which is an excellent data set. And the advantage of that data set is that it really shows where so-called expert pathologists differ in their interpretation of what is actually the TIL and what is actually a fibroblast, and where the pitfalls are for machine learning tools on a very tile-based method. These are three different things. We might consider, but the low to that path is a bit far, but is how can we integrate all these different sets of training materials so that we can develop one publicly available data set that can be used for the community as such. Because probably these three different, they might provide machine learning tools with three different types of information because they have been developed in a different way. So that's a first comment where anybody is free to comment on. The second thing, Brandon, is considering the realities about how drugs get approved these days by the FDA. And if you look at the analytical validity requirements of the companion diagnostics that have been approved, it's extremely poor in content. And my interpretation is that they just being used, the approval route for a drug is being used to approve the companion diagnostic in the same vein. The analytical validity requirements are public, which can be reviewed for all the companion diagnoses are very limited in terms of number of pathologists, in number of cases that have been analyzed. And I do fear, and again, if we think of what will happen with the HER2 low drugs and assays, that we might end up with the same thing. So the Destiny Breast 06 will probably, I think, so considering the biology, will become positive. The drug company will submit a file for FDA approval for the drug, and they will link it to the companion diagnostics used in the trial, and both will be co-approved, and that's it. So I was wondering, would there be another route that we might develop together with the company, together with other organizations that can help fund this grand challenge ideas about HER2 lows, and which we effectively develop together already? If you want to develop or introduce an HER2 low companion diagnostic to the FDA, this is what you have to compile in that file, and together with the community, together with organizations that are willing to coordinate and organize and fund this, together with pathology groups that can provide all the training materials, would this be something that we could start to think together over the next few years before the HER2 low assays get submitted to the FDA?
So I'll state something that is just the way things work is we will probably never require that. When we give guidance, it's about the spirit of what we need to see. It's not about you must use this data set or you must use this figure of merit, this performance result. It is, we need to see a performance result. We need to see an appropriate data set. But if you build it, the drug companies will come. That's my belief. It's hard to turn away from a data set that's sitting there like at the end of the rainbow. Once you've got your algorithm and you've got your drug and there's this data set of outcomes and patient information just sitting there to be used in a regulatory submission. Now, I understand that with drugs, that the business and their phased approach to clinical trials, maybe that doesn't fit into that, but it does fit into the device side of the house. And so in terms of creating the data set for that end purpose, I'm all for it. That's why I'm here. That's why this ICC exists. And my management knows that I'm in the data set generation business, even though it's not really an FDA business creating regulatory data. However, the community knows, and Joe will point to some other real creative point in situations where we do for the whole COVID testing. We created the ultimate evaluation standard test and samples. I didn't know that. I wasn't in that particular technology space, but my belief is, and genome-wide assays, those sorts of things where truth is so hard that we're already doing this sort of thing of working with the community to create not just the reference standards, but the analysis method, because the dimensionality of that problem is just so much bigger. But for biomarkers, I feel like it's such a achievable direction, and once we get these models and algorithms cleared by expert pathologists as the reference standard, in some cases that reference standard is going to be obsolete, and it's going to be the models that are the reference standard, and because these are tedious, counting, measuring, humans are not good at that, and but that's what we have right now. And a lot of when we're talking about target finding targets and counting targets and measuring targets. The expert pathologist is our reference standard until we can get an outcome, and then we don't need the pathologist to look at the image. We just look at what happened to the patient, survive or don't survive.
Yeah, I think this is a great example where, you know, once the one seed has been shown to work, then, you know, everything else will fall in place. I believe that the combination of the high technical difficulties of converting a lab into a digital pathology lab, coupled with the somewhat high challenge of integrating an AI tool into clinical workflows, that's still a high hurdle for, you know, the average pathology laboratory. So I think that creates probably a bit of a delay, but I think what we're outlining here is absolutely the right way.
Yeah, I mean, my research project right now is about getting those, you know, having a controlled study to collect pathologist annotations that could be used in the regulatory setting. You know, I'm talking with Jeroen and his colleagues at Radboud about doing expanding the tools they have for reader studies, and at the same time talking to Kayvon at IDC. I don't know which platform, you know, I probably can't do all platforms, but, you know, and IDC, it's gonna, and probably also with the Grand Challenge, it's about standards and interoperability. So that's a big focus of my goals, and it doesn't mesh really well with FDA goals, is to be worried about standards. I mean, we believe standards are good and great, but we don't invest a lot in those at the FDA because we've got too many other things to spend money that cost money. And you know, and I'm trying to get the help to do that. And fortunately, I have a lot of volunteers and groups that are helping in these little ways that they can to either get me data or get me access to some of these platforms or get me the pathologist to do these annotations. Those are the things that I need, as well as, you know, more statisticians kind of contributing, and you know, the different organizations to help us. We're creating expert continuing education credit training for our pathologists that will be in the study. There will be a proficiency test, and the pathologists will be qualified in some sense to do this TIL assessment, so that there's some reference standard. Because too many times I was in a room and people said, well, what's the qualifications of the pathologists? And I would say board certified. That's all I knew, really. And they're like, is that really good enough? And looking at some of the pilot data I have, not feeling that is good enough. There's a lot of variability there, but the more we have discussed training and talked about the instructions to give to the pathologist, and we're turning this into an actual continuing medical education credit situation, so that not only are we helping our reference standard generation, but we're contributing to the clinical experience, hopefully giving something that pathologists can use in their clinical space. And so all these things swim around together as long as you are open to including them all, because like I said to Roberto early on, it's hard to get them all in up front because the value proposition, the costs, and is really, you know, when you need to do something, you need to walk that line and not get distracted when the person who's paying for it is breathing down your neck. Fortunately, as a government employee at the FDA, I have a little bit of liberty to grow the collaboration group and involve a lot of different viewpoints and try and do it right. So my project is hopefully going to demonstrating some of those things. And I think already in some of the materials we've been sharing and putting out there, some good things to think about. And I'll point out, there was a paper just not too long ago from Wahab in England on what to produce when you're creating one of these data sets. And if Kate or someone could drop that in the the chat before we're done. We're going to try and replicate that, because I think all the points that they talked about, Roberto, you shared the paper with me. It's all about documenting how you created the data set that will be the reference standard. I thought it was a great thing that we were going to reproduce.
So what are the next steps? Because we are almost out of time, and I think this discussion is by no means over. Should we maybe follow up in another discussion after having these points sort of really collected? Because I think there's a lot of very important seeds here.
Joe, can I?
Yeah, go ahead.
Can I propose a use case?
Sure.
Let's think about. There is a real clinical need in the near future, and that's SS for HER2 low. First of all, we don't know what the HER2 low is, and there will be evidence public in the next month or two showing that immunohistochemistry is by far the worst assay available to define what is a HER2 low case. And actually, that you should go, you should look at it with other assays. Let's define with people from this group, together with the TILS working group or experts from that group and the pathology innovative collaborative community and others, how can we define a grand challenge in which we include everything that has been discussed today, go to the company. I have a meeting with them next week on this topic, by the way, to propose them a project to do this type of private-public partnerships, validating using grand challenge platforms, different technologies on the same data set, that we can define later what the data set will be. So that information is available already before the drug company goes to the FDA with their trial-specific assay. And in that way, we might solve the companion diagnostic conundrum that we are having today. Because then the FDA will have a lot of assays available, and then maybe then we might find a rational approach to companion diagnostics in daily practice.
I think that's great. If you have other proposals, please just send them to the PyCC email, and we follow up with the next meeting to discuss that. But I think this is great. Gianna is in. Perfect. HTT updates. I mean, TILS and HER2 low seem to be already two things on the docket, so maybe we can discuss that at the next meeting. All right, let's close this. Almost on time. Thank you so much for your time, and hope to talk to you soon. Bye.
Thank you, Joe. Thanks all.
Bye-bye.
Bye-bye. Great discussion. Bye-bye.